Ashmin to introduce Professor Walker. Professor Walker's talk is co-sponsored by Dr. Amruta Ishwara.
Giving me this opportunity to be here and sharing the excitement with all of you, which is really a wonderful moment that I'm here. I was going to say a few things about biochemistry department, but already Ranga has said a lot, and Anurag said the remaining part, and Dian has also said something. So I think I'll only say that I think the biochemistry department, as we have known, has also been known as the mother department for many of the biology research groups here and the departments in the institute. So congratulations on this centenary. I'm really happy to be here. In this session, there are two speakers, and both of them are very well known to me. The first speaker, ANAND SWARUP, was my senior at Pantnagar, where I did my master's. I joined there in '76, and he would have joined, I think, in '75. He was in the same batch as D.N. Rao. So we've been knowing each other for a very long time, and it's really nice for me to be chairing the session in which he's speaking. Anand SWARUP is a lab chief at NIH. He's a senior investigator, and which one? Oh, sorry. Yeah, I'm sorry. Yeah. Graham is also very well known. I thought you're giving me in this order. Well, I can share a story about Graham because, see, I did my postdoc at MIT, and I joined here in 91, but then when I went back in 91 to visit, we moved to a new building in 94 from the old building, and Graham's lab and Raj Bhandari lab, they were neighboring labs. We were just connected by one door, which was supposed to be kept open actually because of the safety regulations, but it was closed actually. So I think it's really wonderful to see Graham here, and I think this talk, Graham is American Cancer Society professor, HHMI professor, McVicar faculty fellow. He received his PhD from University of Illinois in 1974. He did his postdoctoral work at University of California, Berkeley with Bruce Ames, and this is what I think he's going to begin his talk with. Since then, he has been associated with several societies and academies, American Cancer Society, National Academy of Sciences, Professor Walker studies DNA repair, mutagenesis, and cellular responses to DNA damage, as well as the symbiotic relationship between legumes and nitrogen-fixing bacteria. He's been awarded many honors during his career. Just to name a few: Environmental Mutagen Society EMS Award in 2006, Revolutionizing Innovative Visionary Environment Health Research, R35 Outstanding Investigator Awards, 2017, and so on. He's a member of the National Academy of Sciences. Graham, it's really a pleasure for me to invite you to give this talk in the centenary session here.
Thanks for that very kind intro, Ramesh. We did open the door a little bit later when we started to collaborate on YBY. It's an extraordinary honor for me to be here and join you in this amazing celebration. I have friends who can't believe that you brought me here all this way, and I'm still a little in awe of it myself. I've 100 years. This is amazing, and I've been in awe of what I've heard. On the other hand, I've felt the weight of history on my shoulders as I've thought about how to do this talk, begin this talk for quite a number of months. And I've been at MIT 44 years, and only once before have I sort of felt this weight of history on my shoulder, and that was the time that the Cold Spring Harbor Symposium finally got around to biological responses to DNA damage, and I was asked to open that session, and I didn't know what to do, but I remembered Bruce Alberts, the wonderful biochemist, giving a talk on protein machines at a previous symposium. So I thought I'll look up and see what Bruce did, and that'll give me an idea what to do. And when I did that, I wrote about this in my paper. What Bruce wrote was, when I was asked to prepare this introduction, I very nearly declined, not being nearly as wise as Alan Campbell. I knew I could not attempt to match his scholarly introduction to the 1980 Cold Spring Harbor Symposium. And I thought, I'm not as wise as all of those guys. And I feel that sense again today, but I'll try and do my best. I did have one sort of insight as to how maybe to begin this talk, and that your lovely history you've described stresses the fact there's sort of two phases to the research here. The 50 years of, approximately 50 years of classical biochemistry, and then with the advent of molecular biology and recombinant DNA technology, structured DNA, genetic code, that changed everything, and you got all these other subjects after that. And I was thinking, well, I was a postdoc in Berkeley. Recombinant DNA was invented right across the bay at UCSF and Stanford, and so I got thinking a little bit more about that and my life. And I was 5 years old when Watson and Crick discovered the structure of DNA. But where it entered my life was when I was an undergrad at Carleton University in Ottawa. I was a chemistry major. I thought I was going to do quantum mechanics, but I had to take an introductory biology course, and there was DNA. And I was fascinated by the elegance of the molecule. I realized it was too big for quantum mechanics in the 1960s, so I said I'll become an organic chemist and I'll synthesize DNA. And that was what I did. And that inspired me to, in my next year, take a genetics course. And first of 3 Indian scientists who had a profound impact in my career, V and Bob Iyer at Carleton, taught a course using the molecular biology of the gene, Watson's first book. And that just cemented my enthusiasm for the younger scientists here. I have a page from that book. That was the genetic code. They hadn't finished it when I was an undergrad at that point. They didn't understand it all, just to sort of give you a sense of perspective. And then I, with a wonderful, my undergrad research mentor helped me. He was collaborating with Saran Narang at the National Research Council, and I got to work in Saran's lab. He was honored as one of Canada's greatest scientists, but what he was doing was trying to synthesize the first gene chemically, and he was competing with somebody called Govind Khurana, who was trying to do the same thing. I had no clue at the time that Govind would be my colleague, and not only, he was just down the hall along with Tom Rajmandari and was my close neighbor for many years. I didn't see that coming. Anyway, this background inspired me to go to PhD in Illinois. As you heard, I worked as sort of nucleotide, oligonucleotide biochemistry, and went out to Berkeley to learn genetics, and I'll tell you about that in a minute. But in any case, as a result of that, over my career, I've worked on a variety of these areas that have been studied in the last 50 years here, but that included DNA repair, and that particular area is going to be relevant to what I was going to want to talk about today. But I thought I'd just, to put this in a broader context over this 100-year period, tell you something about how we learned about DNA repair. When Watson and Crick solved the structure and wrote this paper with this famously coy sentence that said, has not escaped our notice. The specific pairing we postulated immediately suggests a copying mechanism for the genetic codes. They realized the 2 strands being complementary, you separate them, you copy each one, and now you got 2. But what they didn't see, if you have the information there twice, you could have damage on one strand and fix it up accurately because you still got the information on the other strand. They did not see that. Phil Hannawald, who's sort of grandfather of DNA repair, says they admitted they didn't see it. But not only that, the whole field didn't see it. We take it for granted now, but that wasn't always the case. It was well known that by this point there were chemicals that could damage DNA. Some were endogenous, like reactive oxygen species. Others were environmental, UV radiation, or abasic sites, pyrimidine dimers, cyclobutane dimers, 6,4 dimers from UV, benzpyrene addicts, or things like that, or say cisplatin crosslinks, for example. And in the particular context of this talk, I'll point out that some of the chemical carcinogens in cigarette smoke are things like that benzpyrene addict, polycyclic aromatics, or melanoma, UV. And how do we treat these things? We treat them with more DNA damaging agents such as cisplatin or cyclophosphamide. So anyway, the community at large did not think that DNA repair existed. It hadn't occurred. So geneticists did screens for more resistant mutants. And with a couple of exceptions that had nothing to do really with DNA repair, but made you resistant to UV or something, they didn't find much. The mutants weren't impressively resistant. And so it was a really breakthrough. In 1958, Ruth Hill isolated the first UV-sensitive strain, and it was really sensitive. And the light bulb went off. Maybe nature had already invented DNA repair. You didn't have to try and make it do it. And if you look for sensitive mutants to things like UV or gamma radiation, you'd find the genes that were involved. Evelyn Witkin and Peggy Lieb in '61 found if they incubated cells without allowing replication, they survived better, suggested enzymatic repair of DNA. The really seminal work was Richard Dick Setlow and Phil Hanawalt in 1964, who, putting their work together, showed that what happens is you remove the lesion, get a gap, and then you get repair replication across that. And then Jim Cleaver made it all more credible by showing the same thing goes on in human cells, that derm and pigmentosum are equivalent to the sensitive mutants you find in bacteria and yeast. So from that, there are now, we know, 3 modes of accurate DNA repair that are based on this principle: nucleotide excision repair that fixes up bulky lesions, base excision repair that tends to take out things like oxidized or methylated bases, and mismatch repair is the same idea, except the cells have to keep track of which is the parental strand, but in all cases you get an excision gap and fill it back in. We've since then learned that, and I'm going to say the Nobel Prize was for working at the biochemistry of those 3 pathways of that type. But since then, we've learned there are other types of DNA repair. There's some things that directly remove such things as methyl groups from DNA by popping them onto another protein or oxidizing them to get them to come off. I think probably all of you know double-strand breaks are bad, and there are 2 dominant modes in nature: homologous recombination and non-homologous end joining for dealing with that. However, there's another way of dealing with damage, and this is important if a lesion doesn't get fixed up before the replication, before it comes along, because the replication machinery is like a fine-tuned racing machine, very fine tolerances, and it tends to jam when it hits the lesion and stop. And so there are 2 pathways for getting around this, one of which is translesion DNA synthesis, which I'll be talking about, and there's a recombinational bypass mechanism too. But the point about DNA tolerance mechanisms is the lesion still present in the newly replicated DNA. If you can replicate your DNA, it can stay alive, and then you can go back and have another try at trying to remove it. And my talk is going to be sort of focusing on, on a strategy where I've tried to build on the basic knowledge we've learned about how mutagenic translesion synthesis occurs, and try to use it in the same spirit as RENU mentioned this morning, applying it in a translational context to improve chemotherapy. So let's see here. This work actually grew to study mutagenesis rather than the study of repair. People were starting to study mutagenesis during the first half of the work here going on in this department. Work with 1928, 33, 42 by Mueller, Stadler, Altenberg, Charlotte Arbach found that these agents that we now know damage DNA, increased mutation rates. But at that time, nobody knew what the genetic material was, so they weren't really studying DNA at that time. It's interesting, in retrospect, Alexander Hollander's work suggested from the action spectrum that UV was a genetic material, but that was the time where people thought DNA was a monotonous tetranucleoside, so that idea didn't get traction. So the real advance didn't come from this. It came later, and the 2015 Lasker award to Evelyn Witkin and Steve Allage really recognizes some part of this process. So Evelyn was a remarkable person. She started at Cold Spring Harbor in 1949. And in 1967, she published 2 papers that were really very prescient. First, she just noticed parallels between the induction of prophage lambda and the induction of filamentous growth after DNA damage, and suggested perhaps there was some kind of regulator that was similar to lambda repressor that would regulate this network. Well, that's what you now know as the SOS response, and Lexay is indeed very much like lambda repressor in how it works. So that was one part. The second paper suggested that an induced function, induced by DNA damage that she'd been studying, that was needed for mutagenesis could possibly be an error-prone polymerase that copied over a lesion and made a mistake and then continued, and that might be the basis of why you got mutations after DNA damage. Up till then, people just thought it was just stumbling and little mistakes by the regular machinery. So that, I think it was that paper that helped inspire some genetic screens that probably would have been counterintuitive before that. So these were screens for cells that didn't mutate after DNA damage. That was a mind-bending idea at that point. And it was first done in yeast by Jeff Lamont and then Chris Lawrence, and this led to the isolation of the Rev 1, 3, and 7 mutants. These are non-mutable with UV and many mutagens, and I'll be talking about them extensively as we go along. A similar screen was carried out shortly afterwards in E. coli, and that led to the isolation of UMUC mutants that were non-mutable with UV and many mutagens. And my entree into this field came when, as you heard, I went to Bruce Ames' lab. Bruce had just developed the Ames test, this test based on some salmonella strains reverting histidine oxytrophs to prototrophy. That became the first widely used short-term test for potential carcinogens or mutagens. And Bruce made a number of clever things, did a number of clever things to make those strains so effective. But one of them involved the introduction of a derivative of a naturally occurring drug resistance plasmid called PKM101. And if you put it in a cell, instead of seeing this number of mutants for that amount of mutagen, methane sulfonate in this case, you got a lot more. And it really made the test useful, and that's when Bruce went public with it. Bruce didn't care what the plasmid was doing, but I thought there must be something interesting on that plasmid that maybe I could learn something about mutagenesis, and I grandly wrote, and it maybe would help me cure cancer or something like that, because that was how you get money in those days. So anyway, one of the things we discovered pretty quickly was that if you put a PKM101 plasmid into a UMUC mutant, it became mutable again. And formally, there are a lot of possible explanations for that. The simplest one is there's an ortholog of some chromosomal gene, of the UMUC gene, on the plasmid. And as I'll show you, that turned out to be the right answer. At the time, Cynthia Kenyon was a first-year or grad student in my lab. And because these genes were thought to be associated with this emerging SOS response, she used a genetic technique to take a promoterless lac and made a lot of random fusions and then screened to see if we've got any fusions that turned on with DNA damage. And we did, and we called them DIN genes. That was the first evidence in any organism that DNA damage turned on the expression of genes, something we take for granted now. And it was an undergrad in the lab at the time, Ann Bagg Brett, who had a first author PNS paper as an undergrad who showed that UMUC was SOS regulated. Ann's been a professor at UC Davis for a lot of years. This is when Steve Allage, who was also a grad student in my lab, early lab at that time, came in, and he cloned both the genes that were needed for mutagenesis off the plasmid, but also the UMUC locus off the chromosome. And that was pretty challenging to do at the time because in Cambridge, they were very worried about the recombinant DNA being dangerous, and so we were operating under a lot of very weird restrictions of what we could do and not do. Anyway, Steve persisted, and as soon as we saw the molecular weights of the products, we could see this idea that the PKM1 just had an ortholog of whatever the UMUC locus was likely to be true. So we then sequenced these operons, and we found that the smaller protein UMUD and MUC-A shared homology with the carboxy terminal ends of LEX-A and lambda repressor, and it's sort of a regulator. It's a whole other story. I'm not going to worry about it today. But what was frustrating to me was that when we looked at the sequence of UMUC and MUC-B, there were no homologs in the database. And I was hoping to get a clue as to their function from that. So I have a quote from a conversation I had with Colin Manoyle, a wonderful scientist. And I said, it's so frustrating. This cloning, we sequenced the UMUC and the PKM1 version. There's nothing like them in the database. And he said, don't be discouraged, Graham. You probably just sequenced the first members of the next superfamily. And I laughed. But over the ensuing years, that was 1985, more and more people started publishing sequences that were clearly members of this family, but nobody knew what they were doing. And then in one explosive year, 1999, suddenly turned out every member of this family was a translesion DNA polymerase. Some of them can go accurately over particular lesions. Paul Ada is famous for being able to copy fairly accurately over thymine, cyclobutane dimers. Coding information is there, but it's distorted, and so it can bend its active site enough to read that information. And in a similar fashion, let me go back here. I got it the wrong way. DIN-B can copy over N2 guanine adducts. The coding information is there, but again, a replicative polymerase can't read it, but the specialized polymerase can. But more relevant for what I want to tell you in the rest of my talk, UMUC, which is required mutagenesis in E. coli, and REV1, which came up as a non-mutable in the yeast screen, required mutagenesis, are both members of this family. The REV3 and REV7 turned out to be a translesion polymerase we call Pol-zeta. REV3 is a member of the B family. It's a huge protein of 3,000 amino acids. REV7 I'll tell you about is a small hormone domain protein. You could think of it as a polymerase accessory protein for the moment. In any case, here's a summary in eukaryotes now of how this REV1, REV3, REV7 gene products can copy over damaged DNA and introduce mutations. So imagine that we had a C at this point, and let's just imagine it's got some kind of damage, so now it's a lesion. That would stall a replicative polymerase, and when that happens, there's a system that takes the processivity clamp pCNA and puts on a specific monoubiquination. And REV1 has elements in its structure which allow it to bind to recognize monoubiquinated pCNA. So it gets recruited to the place where the replication fork got stalled. And then there's this sort of polymerase-like domain, and I will be telling you for much of the talk about this part up here. There's a C-terminal domain of 100 amino acids that has two different interfaces, and you'll hear what they are in a second. So the first thing that has to happen in translesion synthesis is the cell has to put something opposite the lesion. And without going into the details, REV1 is not very good at inserting things. It only can put in C's, and it's not a very good inserter. It helps, sometimes it puts in nucleotide opposite a lesion, but its more important role is very good at recruiting other polymerases that can put it in, such as these three members of the Y family translesion polymerases, Pol-eta, kappa, an iota, and it uses the interface over on this side to recruit those. All of these things have in common a little sequence motif called a Rev1 interacting region, or RIR, and that IRR sequence binds to this, this interface, gets recruited. Then it needs to recruit Pol Zeta to do the extension, and that uses the other interface, a specific recruitment between this interface and Rev7. I'll show you about in molecular detail. So the effect of these recruitments then, we step back and look at it. This enables the cell to now, instead of stalling at that lesion, to then put something opposite the damaged base, and by the time it goes to the next round of replication, if there was a mistake made here, you end up with a mutation here that was caused by that lesion. And I think you can see now, if you block this, the action of this system, then you don't get that mutations, and that's why you've got the non-mutable phenotype. Okay. So what I want to try and do is then tell you sort of, this is building on a lot of decades of work from my lab and other labs, whether the knowledge gained here could be used to try and help treat cancer. I could have put numbers up. Everybody knows it's a big problem. We probably all know people who have cancer. And the trouble is, even with some a few magic bullet treatments, it's still a huge problem, and we don't know how to do things very well. 50% of late-stage cancer patients get genotoxic therapy, and most die from their disease. The numbers are not very good right now, so there's a lot of room for improvement there. And here's part of the problem, that when you do chemotherapy, often you get the sort of thing depicted here. The tumor seems to disappear, looks like it's gone, and then a little while later it will relapse, and just all too often it's resistant not only to the chemotherapeutic agent you used, but to many others as well. So, and this is the obligate first line therapy for most cancer, especially these late stage cancers. And the approach that I'm going to try and tell you about, I think, might improve both the effectiveness of this killing, but also reduce the amount of drug resistance that you could get. And the idea then came from this kind of thinking, which I'd been doing for years while I was doing all this work in bacteria. I never thought I'd ever get to try the idea in mammalian cells. But here it was that when you treat the cancer with chemotherapy, it looks like it's working, and then it reappears and it's resistant. If you read the literature, much of it says, well, cancers are very heterogeneous and you're just selecting for a resistant mutant. And you can see with the perspective that I had from my work, I thought, well, maybe there's another possibility. You're taking these cancer cells and you're trying to kill them off, so you're putting lesions in them everywhere, and you've got translesion polymerases copying over them, and they're mutating the cancer cells like crazy at the very time that you're trying to kill them off. So what would happen if you could block mutagenic translesion synthesis while you're doing chemotherapy? Could you help with this problem? And when my MIT colleague Mike Heeman first joined the lab, I realized he had a lymphoma mouse model that would allow us to address that question. And to his great credit, he was willing to work with a guy who mostly worked with bacteria and yeast to try out the idea. And this is a mouse model of human lymphoma. And the way it works is you can take the lymphoma cells, you can mark them with GFP if you want, inject the cells in the tail vein of a mice. 15 days later, tumors appear. You can treat them with cyclophosphamide, which is one of the frontline agents for treating human lymphomas. They disappear. But then 15 days later, the tumors relapse. About half of them are highly resistant to the drug, and some are partially resistant. And the reason he knows this is he can isolate these tumor cells, grow them in the medium, sort them if he needs to, and then go back and start over here and just test their properties, and that's how he knew that many of them were resistant. So all we did was to repeat the experiment, except we also knocked down the expression of Rev1 and just did the same thing. And in the first round, it didn't make any difference. We didn't expect it would make any difference, whether we had just a vector or we had included a vector that had an shRNA that knocked down Rev1. But when we recovered the relapsed tumors and then did the experiment over, we saw a really striking difference. This is what he usually saw. Some of these tumors were so resistant that despite the fact they were giving them chemotherapy, the mice were dying immediately in 2 days. Some of them were partially resistant. Whereas if we knocked down Rev1, to a first approximation, it looked like we saw the first time. It wasn't perfect and it wasn't a great knockdown, but it encouraged us to think that this might be an interesting direction to go in, and inspired us. This sort of a proof of principle that by inhibiting mutagenic translesion synthesis, you might be able to prevent acquired drug resistance. So, and then we got thinking about the problem of cancers that are intrinsically drug resistant. They're resistant when you start, and they're very hard to treat. And at MIT, a well-known example of that is Tyler Jack's particular has popularized this use of this KRAS P53 mouse model of human non-small cell lung cancer. And it's famous for being resistant to chemotherapy. This is lung tissue. Healthy lung tissue is gray. And even though the mice are getting chemotherapy, you can see the tumor overgrowing the healthy lung tissue. A component of Pol Zeta gives an even bigger response. That might be in part due to the fact that Rev7 also plays roles in the choice of double-strand break repair between homologous and non-homologous joining. We did one more proof of principle. I collaborated with Oman Farrokhzad at the Dana-Farber, who came, interestingly, out of Bob Langer's lab, who you heard about this morning. And what we did was to make nanoparticles that could deliver either siRNAs against Rev1 and Rev3, or those shRNAs and a cisplatin prodrug that could be put in the nanoparticles. And what we saw was striking. This is a human prostate xenograft mouse model, and if you don't do anything or inject saline, these tumors grow very vigorously. If you inject free cisplatin, you can knock back their growth somewhat. If you now include nanoparticles that have siRNAs against Rev1 and Rev3, the tumors grew much more slowly. And if we put the prodrug inside instead of giving free cisplatin, we completely prevented the growth of the tumors as long as we kept watching them. So that all was encouraging. It's a proof of principle anyway that this might be an interesting direction. I mean, all of this is at an early stage, even at the end of my talk. But so anyway, I got thinking, how could you block this pathway? And there's somewhere between 15, 16, 17 DNA polymerases in human cells. They all work basically the same fundamental catalytic mechanism. So trying to block one polymerase activity without having a side effect of something else is hard. So I wondered if instead you could block maybe some key protein-protein interaction that was needed for this system to work. And I've sort of set it up for you that there are a couple of different interfaces on the C-terminus that are important for this process. And indeed, what we've tried to do is block them. But although we had to know something to come up with that idea, and I'll just show you how we found out about the C-terminus and its interfaces. When I was named an American Cancer Society professor in 2002, I'd only worked on E. coli, or bacteria. And so I decided, stimulated that to try and work on eukaryotes. I went to yeast, and I worked on Rev 1, 3, and 7. And there was some emerging evidence in mammalian cells that the Rev 1s had a C-terminal domain involved in recruitment, but it was widely held that yeast did not have that. And when my colleague Eric Lander sequenced a bunch of yeast strains before he published, we got hold of the sequence, and we could immediately see when we lined them up that yeast had a C-terminal domain as well. That we showed genetically it was important for its function, and we predicted that it would be 4 amphipathic helices would constitute this recruitment domain. And I then collaborated with 2 different structural biology groups, one of whom did the mouse C-terminal domain of Rev 1, and the other did the C-terminal domain of human Rev 1. They both, not surprisingly, found the same structure. These 4 amphipathic helices formed to make an atypical helix bundle, and there were 2. We worked to show that there were 2 interfaces. The one here binds the Rev 1 interacting, or RIR regions of Pol eta, kappa, and iota, and the interface up here interacts with the with the Rev 7 component of Pol zeta. So these are the recruitment of the inserter polymerases. This is the recruitment of the extender polymerase of Rev 1. So no sooner had each of those groups solved the structure by NMR. Sorry, I meant to say who the groups were. It was Dmitry Korzunov at University of Connecticut and Peju at Duke led the 2 groups. And no sooner had each group solved it by NMR than Pei managed to get the crystal structure of a quaternary structure that had the Rev 1 C terminus, had the little Rev 1 interacting region from Pol kappa. It had Rev 7, which is the component of Pol zeta, and a little piece of Rev 3 that I'll get to in just a minute. But in any case, what was so significant about the structure was it had the 2 interfaces that we were interested in. This is the interface involved in recruiting the inserter polymerases. This is the interface involved in recruiting the extender polymerase, Pol zeta. So we've collaborated with Dmitry Korzunov, who's the structural biologist, Carl Hayden, who's good at doing small molecules. And we found inhibitors of this interface that recruits Pol eta, kappa, and iota. And they found by screening through a fluorescence polarization screen. They've done some virtual screens since, and we're beginning to get a number of small molecules. But the first ones we characterized that interfere with this recruitment were shown here. By adding these small molecules to the protein, we could see by NMR they were affecting the residues right at the interface. So we were sure they were binding where we expected they would. And then we looked to see if they had biological activity. So what's happening in these experiments is we're just treating with the compounds by themselves, maybe a little effect on growth, a low dose of cisplatin, but then if we combine either of these compounds, we saw a substantial increase in killing, suggesting, maybe not too surprisingly, that we could improve the killing with this kind of compound. Just to keep in the back of your minds, we noticed a lot of apoptosis, increase in apoptosis when we did this. That's not a surprise. You see apoptosis with free cisplatin. We weren't sure whether we'd affect mutagenesis, but in fact, we did see a lowering of the amount of mutagenesis induced by cisplatin when we included either of these compounds. And possibly the reason why is Pol zeta is a little more complicated than I've let on at this point. It not only has Rev 3 and Rev 7, it has 2 subunits that are also components of the replicative polymerase Pol delta. And this component has one of these Rev1 interacting regions that goes to that interface. So even though we're probably very effectively getting rid of the recruitment of this, we may be also affecting the recruitment of the extender polymerase as well. One last kind of compound we've made, just came about from knowledge of the nature of the Rev1 interacting region. This was his consensus sequence. It was kind of puzzling at first. A couple of phenylalanines in a sequence that couldn't be proline, but could be anything else. Well, it turned out the two phenylalanines go into pockets on that interface on the Rev1 terminus, and the rest are dominantly backbone contacts, so it just needed to be an alpha helix. So what we have done then is to use a hydrocarbon staple to lock this thing into an alpha helix. And because there's a little more room here, we've replaced the phenylalanines by cyclohexylalanine. This thing works just about as, in fact, it's working probably a little better than most of the small molecules we have. But all of them are blocking this, this interface. I will say, though, when I started this, the one I really wanted to try to block was this one, because I was pretty sure if we could disrupt the interaction between Rev1 and Pol Zeta, we might really have a big effect on mutagenesis as well. And this work was done with Peju. We both screened for compounds, both got some hits, but he got one that looked so good, we just dropped everything else and focused on a compound called JHRE06. I may just call it JH. And what it does is it acts on this interface. There's the interface I showed you before. And I want to now tell you something about how this interface works. It's between the Rev1 C-terminus, you've seen its interface, and Rev7. If you don't know Rev7, it's an interesting protein. It's a member of the Hormad family of proteins that has a C-terminus that can be disordered, but it's capable of binding around a peptide from another protein. In this case, it's some part of the polypeptide chain of the Rev3 polymerase, and it makes a buckle. So this is called a seatbelt, and this is the buckle shown here. And the residues that interact with the Rev1 C-terminus are located on this buckle of Rev7. So it's these residues here, these three residues. And what they do is they poke into this interface right here. To show you that in slightly more detail, here's the interface on the Rev1 C-terminus. It's sort of a little greasy ring, some leucines, tyrosine, and then the three residues from Rev7 poke into the middle of that hole. So I was excited because I thought, well, that's a small enough protein interface. You might plausibly be able to find small molecules that could bind in there. There's not that big an interaction surface. There was a potential problem. Quite a few experts warned us, and me in particular, that that was too shallow, and you never get enough energetics, enough binding of a small molecule to achieve the purpose. But anyway, it turned out we were able to get compounds, and I'll show you the trick why it seemed to work, at least in the case of the molecule I've mentioned. Here is a compound that binds to that site and breaks that recruitment of Pol Zeta by Rev7. The structure isn't terribly important, but the only thing I want you to notice is it's asymmetric, okay? And it's just slightly submicromolar. But when we did the NMR experiment, we saw that expected residues are being affected. So Peju then decided to do the crystal structure so we could really see where it bound, and we got quite a surprise. That molecule bound to the interface, same interface of two different Rev1 C-termini, causing them to dimerize. And if you look at a space filling, you can see how the practically buries the small molecule. And we think this is, because it's asymmetric and it's binding to the same site, that means it has to use two completely different set of molecular interactions in order to bind there. And the interaction with one are shown in yellow, and with the other C-terminal domain in blue. I told Pei when he called to tell me this news, I said, it's like you've won the lottery twice with one ticket to get that. And we don't think it's a crystal packing artifact, because we try to cross-link. In the absence of the drug, we don't see the C-termini cross-linking, but if we add it, then we can cross-link them. So I think a phenomenon occurs in solution. So what that suggests then is the way this drug works is it takes two Rev1 C-termini and sticks them together, and because of that, they're not available to go and recruit Rev7. That's kind of what we are thinking. We asked, well, do they work biologically? So we have four different tumor lines from mice and humans, and each case, very little effect to the drug alone. A low dose of cisplatin, but I think you can see in every case when we included this compound, we got a really big increase in killing. If we looked at mutagenesis as well, here's the mutagenesis induced by cisplatin, and we suppressed that below what we would see even with the drug itself. So that was very, very encouraging. That was the kind of molecule I've been dreaming about. We have evidence that the effects are Rev1 specific, because we have some mouse embryonic fibroblasts that are either Rev1 plus or Rev1 minus. And if in the Rev1 plus version, the small molecule increases the killing. If the cells lack Rev1 in the first place, it doesn't do anything. And if we take this strain, this cell line, and we restore Rev1 through a plasmid, we see back the sensitivity. So I think the effects on killing are Rev1 specific, and the same story goes for mutagenesis, that it suppresses mutagenesis in the Rev1 case, but if you're Rev1 minus, it doesn't matter, you inhibit it. So I think it seems to be acting, I think, by direct effects on Rev1. We did a xenograph experiment, and I think just the pictures probably show what's going on. If we had a combination of cisplatin plus this drug, we essentially blocked the growth of the tumors, and the mice lived quite a bit longer. So, I mean, that's where we're at in terms of this. We have a lot more to do. What we're excited about is we have some indication that the cells are dying after, I'm about a minute and a half away, is that okay? Away, dying from some mechanism other than apoptosis. There's Bob Langer and his colleague Oliver Jonas made a little device. It's a little tiny cylinder of plastic, and you can put different combinations of drugs, implant it in the tumor, pull it out, and then section, and you can look at each little emitter point and see what's going on. And what was surprising was with cisplatin, you see a proliferation marker gets knocked down, more of it when you add the compound, but at the same time, cisplatin alone, you get a lot of apoptotic cells, and we stopped seeing them, and instead we saw a marker that's normally associated with senescence. And when we looked for senescence associated with beta-galactosidase, which is a more widely used marker, we could see it. So at the moment, up to the day I left, we're looking at a puzzle. We're seeing a lot of observations. The death mechanism isn't mapping on top of anything that we know about at the moment, and in particular, they're dying by something other than apoptosis. So we're quite excited about this. I just want to thank, I've named all my collaborators. Most of the work, recent work, was done by Nimrat Chatterjee and an undergrad, Abby Harris. And just to close this out, Bruce Alberts didn't help me get this started, but when I was doing the Cold Spring Harbor, I said he helped me finish it, because I read his closing, and he said a task seemed unimaginable to me at the time of the 1968 Cold Spring Harbor Symposium. Now sees realistic goal. And he says, this is because we're getting all this new technology and everything. So I said, well, you know, can't imagine what it'll be in 16 more years. Well, that was 2016. That seems like old news. So I think you need to think more broadly in a case like this. So congratulations on your centenary. And I feel sure if I could be here in 2021 that the science you'd hear about is just going to take your breath away. So keep at it. Thank you.
Beautiful talk. Can we take a couple of questions? Yeah, okay. In the cisplatin crossing, does the chirality of the crossing, GG inter versus GG intra, determine the Rev 7, Rev 1, Rev 7?
I wish I knew the answer to that. To be fair at this point, many of you may know this. Cisplatin is famous for making cross-links, although it makes a lot of monoadducts, and it can do 1,2 and 1,3 adducts and stuff. And at this point, we haven't sorted that out. And it could be purely translesion synthesis over a monoadduct, or in those of you who know the details, cross-link repair is a problem because you've got things on both sides. That mechanism of cross-link repair also engages the translesion synthesis mechanism. That's all work, future work we need to sort out.
This sort of follow-up might be politically incorrect. In the honor list DNA repair, I didn't find the name of Howard Flanders along with Anna Walt and Bruce Setlow. Is there a reason?
So whose name?
Paul Howard Flanders?
Well, I didn't go into, I mean, I was staying away from that end of things. I think Paul was important in the Rec A stuff. He was kind of absolutely against the SOS idea. And so he ended up, the way I ended up telling the story, in fact, when I interviewed at Yale, he said, hi, I'm Paul Howard Flanders. I don't believe in SOS. Just before my talk, that wasn't a very good start. I got to be friends with him, but.
So what's the concentration of these molecules work? Is it nanomolar, micromolar?
That's just submicromolar. I mean, this is the hit. No improvements. Peju is making more compounds. We had some hits that we haven't followed up on. One in my lists also I'd like to try. As I know that the C-terminus itself is a strong dominant inhibitor. It makes the cells like a null mutant. So if you could sneak in that 100 amino acid, that might be a drug too, that domain. So that's an idea we're about to try.
And it becomes a lot more cytotoxic. When you do the treatment with this compound that you have, does it become a lot more cytotoxic?
We haven't seen very much cytotoxicity associated with it. What we are realizing is if you make cells Rev1 minus, we've gone back to like CRISPR knockouts, and we realize that the Rev1 mutants are a lot worse off than you'd think from the publications in the literature. So there might be some effects on growth, but that'd be just mimicking what happens in Rev1. But there's a Rev1 mouse that Niels de Vin made, published it in 2006, and the wild-type mouse is about this big, and this is about that big. So I think there might be some of the effects, and maybe what happens in vivo may be a little bit different when the cells are on a plate and maybe getting under oxidative stress and stuff the way we grow cells. So there's a whole series of experiments we're thinking about.
Okay, if there are no more. Okay, there's a question in the back. Maxima.
Yeah, it was intriguing to see that the small molecule was inducing dimerization of some kind. Did they have a propensity to be dimers? For example, some homologs in other species were dimers and this enhanced it, or was it completely unexpected?
It caught us completely by surprise. There'd been no indication at all up till then. On the other hand, I mean, all of you know there's a tremendous amount of excitement over these liquid-liquid phase separations and bimolecular condensates, and it's not inconceivable that where these proteins are acting, they're at a higher concentration than we would guess they would from counting numbers of molecules in a cell. But I don't know. you're not the first person to perhaps thought in the back of your mind that maybe this molecule didn't invent the dimerization. Maybe it's something nature does. We just didn't know about it, and it's encouraging them to stick together. But I don't know the answer to your question yet, but it's a good one.
Okay, I'd like to thank Graham once again for wonderful talk and something that you started out that you were just thinking about cancer. You've actually, I want to tell what Graham actually completed
His story, that not only that the door was opened, but actually Graham became a ribosome biologist. This is, I've already spoken about Anand, told you about things about him. Anand obtained his PhD degree in biochemistry department, so he's alumni of this department. He completed his postdoctoral training at Yale University in molecular biology and human genetics. In 2007, he established the Neurobiology, Neurodegeneration and Repair Laboratory at National Eye Institute to advance research in all aspects of retinal biology, disease, and therapy. The primary goal of his lab is to elucidate molecular and cellular pathways underlying retinal development, aging, and disease, and to design novel treatment modalities for blinding retinal diseases. Throughout his career, he has been honored with several awards, including the Board of Directors Award from the Foundation Fighting Blindness, the Harrington Senior Scientific Award from Research to Prevent Blindness. Anand, it's a pleasure for me to invite you to give the lecture.
Thank you very much for the invitation, and it's sort of rather humbling experience for me to be here. I was a student here, as you know, and a lot of my mentors, my professors are here. I heard the history of this department, which sort of was really illuminating. I didn't know a lot of things about the department, so it's been really amazing to be here. What I'm going to do today is tell you my story, how I began in this department. As it was pointed out earlier, it's time for reflection. No, no. My voice is loud enough, I think, even the last person can hear. Oh, okay, now it's, okay, great. Yeah, so I think it's time for reflection on one hand, but also time to look for the future. So what I'm going to do is I'm going to sort of start where I began, and then I will tell you a little bit about some of the new studies that we have begun to do that sort of bring us back to where we started. 3 or 4 years ago, when I was here at Professor Ram Sharma's 50 years at the department, I didn't have that story because there was no mitochondria. I didn't want to tell him some of the stuff that we were doing, so I think some of that will be a surprise for him. So I would like to dedicate this to all my professors, where I learned basic biochemistry, a little bit of molecular biology, and specifically, I think I have to thank Professor Ram Sharma, where it all began. And I think one thing which I learned from him more than anything else was his passion for and his love for whatever he does, and love for mitochondria. I don't think I caught up that very much at that time, but I'm coming back to it maybe after 40 years. So you'll have to, I'm a little slow learner. I also would like to particularly remember Professor Kurup and Professor Padiati, who were very much instrumental in my early stages, and I sort of will miss them, and Professor Shastri. But I'm going to sort of show you some of the pictures. Let's see. No? Am I going forward? No? It's not moving.
You have to push just a little bit harder.
No? Okay, all right, the middle one. All right. Okay, 4 of us in 1977, we came from Pantnagar and we joined IISc. We didn't know, we were 20 years old, most of us. Professor D.N. Rao. I have a bunch of his pictures, but he threatened me that I shouldn't show anything too embarrassing. And I can't do that because he's the one who invited me. So thank you. But I think I won't show that, but I'll show some of your pictures. I started working on mitochondria in Professor Ram Sharma's lab under stress conditions. You can see heat stress and the cold stress, right? You could see this. And it was really stressful. Not really. I think mitochondrial work was the easiest one. And you had to start in the morning at 9, 10 o'clock. Muda and bunch of other people will prepare the mice for, or the rats for you, and you sort of do the experiment. By 5 o'clock you are done, because the mitochondria will be dead anyway by then. So it was great, and you can go to movies, talk to your friends. So it was not really that stressful as it was pointed out earlier, but we used to still stay for 12 hours. Why? Because we had wonderful friends and we had a lot of fun. We used to talk science actually. Oh, actually let me go back. I don't have some old pictures of Professor TR, so I'm going to show you my family, and I have sort of had pleasure of having him at our home, but this was, I think, at his house. This was in the lab after I came with my kids when they were young, and I was showing them where I used to work. That was my bench over there. This is with Mrs. Ram Sharma and my kids who were young at that time. I had full head of hair. You can see that. It was wonderful, and I was very productive. Despite not working as hard as some of you probably do, I had 7 papers, and I finished in 5 years, a little less than 5 years, and they were in good journals. I learned a lot. I learned a lot from him, from Professor Kuru, but also from my friends. This is the picture of 1981 Biochemical Society. I was the secretary of that, and the politics of that the election was all started in microbiology actually, and my guru Nagaraj is over there. He was instrumental in sort of creating all that politics, but it was sort of so we call him Guru. He also corrupted many of us. Some of us who came, we had no idea. So I don't want to embarrass DN because he invited me, but Guru, you didn't invite me, so I can do that. So he was the one who took me for the first time for beer drinking, right, on Brigade Road. So that was great. So that's where we used to spend time. Some of you might recognize some of the people here. I don't know, this is Harin Basavda. There are a bunch of people from our group. We used to go for picnics and all. Microbiology and biochemistry in those days used to be very much tied together in various ways. The secretary was Krishna Kesari, my very close friend who passed away sadly. But we used to have a lot of those picnics together. And there are some other pictures here. You can see Naeem is here. I saw him after some 40 years or 35 years. He's over there in that picture here. And there are a bunch of other folks that you might remember. And this was in 2008 when I visited, pictures in the old department. And again, I came to my old lab. This is with DN again. This was my son. A few years later, I brought him to show where this whole place, where I studied. And there are some more pictures here. Here you can see Umesh. That was a few years ago. And Nagaraj and DN, of course, and a bunch of people from our batch. And DN, of course, I see him quite often. This is in Maryland with my wife Durga and Prasad, who were also alumni of this department. So all that is great to remember. And but this was the group, 77 group. There are few of us are pictured here, but there are many more. George Thomas and many others who were part of this gang in 1974. Vijaya, you see here. Oh, let me go back. No. Yes, you can see Vijaya here, right? And Ajit and Ramana Murthy and other people here, Harin Basavda. So, and we had many others. So we had a great group of people at that time. And we used to meet sort of every week. We talked about journal club. So we used to sort of run to the library, who is going to get the latest paper from Cell or Nature. Even though I was working on mitochondria, I used to be selecting paper for molecular biology. And I used to be stressed out, what am I going to do after I finish this work? And that's where I was just getting crazy for molecular biology. I needed to clone genes. Okay, 1977 to 82, that was the time Tom Maniatis had cloned alcohol dehydrogenase, and libraries were being made, and we were presenting those papers, and I said, I don't know, I need to make. And Gopinathan, Professor KPG, sort of recommended me to someone named Alan Garan. And of course, I became a geneticist. I went to do Drosophila genetics. Then I realized I'm a little bit colorblind. I'm a deuteranop, so I cannot see red and green as well as many other people can. So I decided to forget about Drosophila. I thought I should work with some systems that are where you don't need to know much color. That's the human genetics. So from Drosophila genetics, where I cloned my first gene, P150 GLUT, which turned out to be pretty well known, it's a dianectin in a heavy chain. I went to do human genetics to clone Duchenne muscular dystrophy. But somebody else scooped me, and so we were I was worried, what the heck I'm going to do now, my life is over, since somebody else already got the gene. Next to Duchenne, there was gene for retinitis pigmentosa, and I had absolutely no idea. But I wanted to clone the gene. And my boss at that time, Uta Franke, said, why don't you work on cancer? I said, nope, I'm going to clone a disease gene. So I go to Sherman Weisman's lab, and I said, I want to clone this eye disease gene. And he said, I don't know anything about eye. As long as you're doing something interesting, you can do it. So I became a retinal or eye geneticist. And over the years, for last probably 25 years or so, I didn't do anything on mitochondria. But then, from genes, and we cloned many disease genes, I mean, some 10, 15, 20, I don't count them anymore. But when you go from genes to biology, and suddenly it sort of began to happen, many roads actually, not every road, several roads were leading to mitochondria. And last 3 or 4 years, sort of mitochondria biology has become, again, a big part of my lab. And that's the story I'm going to talk to you about. But if you sleep after the next slide, you would have learned everything about what I really do in my lab. About 4 or 5 years ago, so what I really want to do is I want to tell you the big picture goals. I want to understand how healthy and disease phenotypes are created. Why are we so different? Some of us have sort of no hair, some have hair, some have nose a little fat, some is little long. Why and what creates those phenotypes, those healthy phenotypes and also disease phenotypes? Several years ago, I was part of this consortium called 1000 Genome Consortium, and this paper in Nature had something like 800 authors, so I was one of those 800 authors. But when you read this paper for the first time described the sequence of over 2,500 individuals from many different populations and ethnicities, and identified 88 million, but now it's a lot more. I mean, this is the old story. 5 years is a long time in genetics. So basically what they found was there were just millions and millions of these variations in our genome. And most of these variations are benign. They don't do anything. But some of them really affect the phenotypes. Not only the healthy phenotype, but also there are variations that may lead to what we call disease. So what is a disease? Disease is really aberrant manifestation of the function of that particular gene. I mean, something, and then you go to a doctor and you say, oh, I can't see, or I'm having cardiac issues, or whatever. But what about color blindness? Is that a disease? Or aging, is that disease? So sometimes it's very hard to define what is healthy phenotype or a disease phenotype, but all of these, to some or large extent, is dictated by genetic variations. So that's what my lab does. My lab is trying to understand how genetic variations change phenotypes. But to understand that, you need to really have a systems approach. As a biochemist, you will look at a protein, or a molecular biologist, you want to clone the gene. But now with the whole lot of computational tools, you can do tons of studies. You can create networks, and then you can try to use these networks to understand cellular function or cellular organization. So you don't just focus on genes, but you come a full circle to understand the phenotype. And in my case, we focus on the retina. And And we do all of these things, from genetics to biochemistry to cell biology to physiology, all of these things. And we really would like to understand how these different retinal phenotypes are created. Since most of you do not know what retina might be, or where it is, or how it looks like, I just want to tell you that light enters your, this is the cross section of the human eye. Retina is at the back of the eye, say part of the central nervous system. Light enters through lens and cornea. These optical elements get focused here. And there is tiny little region here with pit-like structure called fovea. Area around the fovea is called macula. If the cells there degenerate, you get what is called macular degeneration, and you lose central vision. And if you lose the photoreceptor cells in the peripheral retina, that is called retinitis pigmentosa. So that's the basic sort of biology that you need to understand. And my lab is very interested in learning how this retina or cells in the retina originate, what happens to these cells in aging and in disease conditions, and what can we do to repair them. So these are the different cell types in the retina. There are photoreceptors here, and light actually comes from top. So it has to go through the whole retina before the light gets captured by the visual pigments in these photoreceptors. Photoreceptors send this information to these inner retina neurons. This is very, very complex. There are three major types: bipolar cells, horizontal cells, and amacrine cells. They process that information. They sort of organize that information in some packets, and that processed information then goes to ganglion cells. Axons of these ganglion cells form optic nerve, which takes information to brain for processing, for you to see. But my lab has focused primarily on photoreceptors. Why? Because they are less complex. So I thought after, sort of when I began to work on photoreceptors, I thought after a couple of years I'll be done, and so I'll move on to inner retina. But for the last 30 plus years I've been focusing only on photoreceptors, and I don't think I understand too much more than what I used to know at that time. So photoreceptors, there are two types. Rod photoreceptors, these are these long cells. They allow you to see in dim light. Cone photoreceptors allow you to see during the daytime, and also they provide you color vision. There are three kinds of cones in humans: long wavelength, short wavelength, and medium wavelength. They have different kinds of visual pigments, and they have different, so they allow you to differentiate between different colors, and I have some defects in my red and green pigments, which is okay. And 7% of males actually carry those defects. Rods, there are only single types, and the visual pigment there is called rhodopsin, and here you have S-opsin, M-opsin, and L-opsin. They have very intimate relationship with pigment epithelium, and they shed 10% of these structures called outer segment. They shed every day, okay, and they are renewed. So photoreceptors are post-mitotic cells, and the reason I'm giving you this information is I'm going to come back to Professor Walker's talk earlier. These photoreceptors are post-mitotic neurons. They are there throughout your life. If they die, you go blind. No cure right now, at least at this stage, but they are highly metabolically active. They consume more oxygen than any other cell in your body, and they have what we call aerobic glycolysis. Even under aerobic conditions, when oxygen is there, they have high amount of glycolysis, and these cells, 10% of these outer segments, these are membrane discs, and they have to be renewed in precise manner. So gene regulation is extremely stringent. Protein synthesis has to be extremely accurate. Any problem anywhere, you synthesize more protein, less protein, you go blind. There is no cure. Now these cells are the only cells in your body that are exposed to, under normal circumstances, exposed to damage. We talk about chemotoxicity and all that. That happens only when you take drugs. But in the normal circumstances, the cell that is exposed to UV is the photoreceptor. And despite all this UV toxicity, there are absolutely no cancers of photoreceptors. Retinoblastoma is a developmental tumor, but in somatic cells, photoreceptor will rather die than get cancer. So that's amazing. Either their DNA repair mechanism is very good, they may have some very unusual, I learned new term, trans-lesion polymerases. I'm sorry, I didn't sort of know about those. I have been seeing Paul Zeta and all these different kinds of polymerases, but I never paid too much attention. I will go back and look at the gene profiles and see maybe there are some weird kind of polymerases that repair these. But one thing is true, they have very, very high expression of DNA repair enzymes, ATMs, ATRs, and many other ERCC6 and other stuff is very highly expressed. And they also not only repair very well, but even DNA methylation there is extremely accurate. So you don't want your photoreceptors to die because you're going to go blind. There's no cure right now. So my lab focuses on all aspects of photoreceptor biology or retinal biology, from cell fate and morphogenesis to see how these cells connect to other neurons, what happens in diseases, and what happens in aging. And we work on age-related macular degeneration, a lot of genetics, a lot of different kind of things, and I'm not going to mention all of that. But what I'm going to tell you is this is how beautiful they look like. These, you can see them. Look at these cones. I think I went a bit too far. So this is a highly laminated structure, and you can see this is pigment epithelium. This is outer nuclear layer of photoreceptors. These are inner retina neurons. Ganglion cells are over here. These cones, when you stain them, you can see outer segments, nuclei, synaptic areas. These are rods. Beautiful structures. You can, this is mouse retina. Now, my lab several years ago identified, when I had my own lab at Michigan, where I was for 18 years, we identified this gene called NRL, which is neural retina leucine zipper gene. Now, this belonged to a family of proteins called MAF family. At that time, there was only one MAF oncogene, but now there are many different kind of MAF proteins. So what we showed was, this young man in my lab, Alan Mears, what he showed was that if you knock out NRL, there are absolutely no rod photoreceptors. So the reason I was belaboring the point, giving you all the background, was I wanted to understand what we are doing, right? I want to understand why we focus on mitochondria. I wanted you to know. So this young man showed that if you look at the mouse retina, these are all the nice nuclei, these are rod cells, inner segment, outer segment. Now you knock this out, all of these are cone cells. Cone cells have very unique nucleus. They have there's a chromatin which is really decondensed. Rods have very condensed chromatin, heterochromatin really in the center, whereas cone is very different, and they have very shorter outer segments as well. This squiggly little thing shows you the function. Dark adapted ERG looks at rod function. Light adapted ERG looks at cone function. You can see this is in wild type mouse. You have very nice rod function. Don't worry about the details. But in knockout, there is nothing. Absolutely straight line. Whereas in cones, now you have very, very high this electroretinogram, big function. Whereas in the normal retina, very, very small cone function, because cones are only 3% of all photoreceptors. One gene just changed completely the phenotype. Now, there was a student in the lab at that time, Ed O, graduate student. What he did was he said, if NRL is required for rod differentiation, is this sufficient? So what he did was he took the promoter of a transcription factor called CRX, and that sort of factor is expressed both in rods and cones before their fate is fixed. So he took the CRX promoter and he expressed NRL, and he found that all cones became rods. And as you can see here, in wild type, rhodopsin is for rods, cone arrestin is for cones. Now you put this transgene, they're all rods, no cones. In NRL knockout retina, there are no rods, all cones. And now you put this transgene, they go back and they become rods. So now we have mouse which has only rod photoreceptors or only cone photoreceptors depending on just one gene. A friend of mine, Douglas Forrest, identified another gene called TR beta 2. He put TR beta 2 at NRL gene locus, and showed now the mouse has absolutely no rods because there is no NRL, but TR beta 2 is responsible for M cone differentiation. Whereas if you knock out TR beta 2 and NRL, you have only S cone or short wavelength cone. So now we could make a mouse which have short wavelength cones or medium wavelength cone or just pure rods. Mice don't have long wavelength cone. So 2 transcription factor could just change the fate. Now you can imagine how many different kinds of projects you can generate. Hundreds of graduate students can take every gene and try to figure out what's going on, right? But we gave up some of those to our students, and some of them are doing other projects. But here is the network we began to build several years ago in one of the reviews, where we proposed that a retinal progenitor cells under the control of OTX2, which is a homeodomain protein, can commit to photoreceptor fate. If you have NRL, these photoreceptors go to make rod. If you have no NRL, they go through a default pathway and make short wavelength cone. And if you have TR beta 2, they become M cone. All right. So NRL is dominant, TR beta 2 is not. If you have one copy of each gene, you make a rod. So you can now change the fate. Now just remember that with this kind of data, you can really do all sorts of things. And what did we do? What we did was, another postdoc in the lab used the promoter of NRL to express GFP, green fluorescent protein, and now he could label the rods as soon as these cells are born. And these cells now you can purify. You can just do flow sort, and now you have rods as soon as they are born, and then it takes them 3 weeks to differentiate to mature. So now you can look at every day or every 2 days you can purify these cells and see how their gene profile is changing, how their proteome is changing, how their metabolome is changing, how their function is getting established. So you can do complete developmental biology now for rods, and you can also mate these GFP mice with another knockout mice. You can now do the same studies with cones, because you can label now those rods that were, those cells that were supposed to be rods now became short wavelength cones, and now you can look at those cones. So this is what 2 other postdocs did in the lab, Hyunjin Yang and Junwoo Kim, 2 Korean postdocs, and what they did was they mated these and the gene profiles at multiple stages of rod and cone differentiation. You can see this is a PCA plot that we have. So here you can look at profile at P2, P4. P stands for postnatal day after birth. Rods are born primarily after birth in mice. So you can look at how the gene profile is going, and if you don't have NRL, the gene profile is same at P2, but then it changes. It takes a different trajectory. So blue here is no expression, red is very high expression. This is wild type mouse, purified cells. P6 to P10, there's a major transition, but that transition won't happen if you don't have NRL. So you can see these cells continue to express a bunch of these genes. These are S-cone genes, and they need to be suppressed in rods, whereas the rod genes are no longer expressed in the absence of NRL. This led to a lot of projects, because we showed that S-cone not only is a default path, but during evolution in mammals, probably S-cones were converted to rods when nocturnality evolved. So mammals, so most of the animals before mammals had cone-rich retina, whereas nocturnality evolved about 200 million years ago or so. And during Jurassic period, when dinosaurs were there, all niches were taken over by these big animals everywhere, water and earth. So only thing that a mammal has to do is survive is in dark. To see in dark, you need rod photoreceptors. And for that, probably nature converted one of the genes called what we call NRL to become rod-specific gene. So that was very exciting for us to correlate that to evolution. But also you could purify rods, and you could do transplantation, you could do and you can build these networks. But these networks are not just for theory, because now you can ask the question, what happens if there is a mutation. So in aging, what happens? Will this network look like this, or will it change in any way? How come one mutation is going to alter the whole set of networks? This is what we begin to do. So now I'll come to degeneration very quickly. I told you about photoreceptors. This is the macular, this is the fovea, macular region, and in macular degeneration, you cannot see, you don't have central vision, you cannot drive, you cannot read, whereas in retinitis pigmentosa, photoreceptors die in the peripheral area, so you have tunnel vision, whereas in macular degeneration, you lose your central vision. There are many, many genes that have been identified, over 200 genes for Mendelian diseases, but then you have much more complex multifactorial diseases where genes and environment interact. So there are over 200 genes for Mendelian forms of retinal degeneration. Now, and these genes belong to all sorts of pathways. So if you want to design a treatment, just forget about the biology now. Now just think about these 200 different genes participate in different pathways, but ultimately, if you have a mutation in one of these genes, it will lead to photoreceptor cell death, correct? So if you want to design some sort of treatment which is applicable to multiple diseases, you'll need to think of pathways and networks, right? Because somehow these mutations are going to lead eventually to photoreceptor cell death. And there are many different pathways along the way. So we thought, if that is the case, we proposed that there must be some sort of common pathways that maybe all of these mutations trigger some sort of, they converge at some place and they trigger some apoptotic pathway by upregulating or downregulating certain genes, because the apoptosis or cell that takes place only by few sort of select set of pathways. So if we focus on very, very early stages, before the degeneration is visible, we might be able to find some pathways and understand where these pathways are converging, and maybe those will be good targets for drug discovery. So just think about the networks that I was showing you. If you have one node which is defective, this can change this whole network. If we can find in how many different mutations this same node is affected, then this would be a good target for drug discovery. So this is what we begin to do. And a couple of students, they focused on very early stages. Again, I showed you this kind of thing earlier. They focused from P2 to P10, and they made transcriptome profiles like we did earlier, but they also did proteomic profiles and metabolomic profiles. They did everything. And these are just some of the mutants. RD1 is a mouse mutant. All of these mouse mutants are for genes which also lead to retinal degeneration in humans. So you can see how fast in RD1 degeneration happens. This is RD1 at P12, photoreceptors are going, you can see, and RD16 is fast, RP grip is even, this is at P60. So you could take all of these, but rather than focusing when the cells start dying, we wanted to focus at very, very early stage. So when we did the transcriptome profiling, okay, now let me come back to mitochondria. Just to give you a little bit of background here, photoreceptors can utilize only glucose. They cannot utilize fatty acids as substrate. Actually, most neurons use only glucose, and they have very high aerobic glycosis, just like cancer cells. And this was an observation by Otto Warburg long time ago. And this is how they look, this is how they are organized. These are the mitochondria in photoreceptors. And this is the nuclear layer, and this is at the base of the outer segment, tons of mitochondria. And then the synaptic area has got single large mitochondria. So complete neurotransmission is dependent upon this single mitochondria in rod photoreceptors. Okay? So you can see very nicely organized mitochondria within these photoreceptors. So when we did a transcriptome analysis, we means Coco Jiang and Anupam Mondal. So Anupam is a bioinformatics person from IGIB and Coco came from Toronto. So they did both transcriptomics and proteomic studies, and what they found was something very striking. What they found was a lot of these OX-FOS genes, I have to look at this every time where I'm pointing, otherwise I can move forward. So if you look at this, a lot of these genes, these are all, many of these are sort of complex 1 subunits and all, and you can see they're changing. I mean, forget about all the just trust what I'm saying. All right. So the bottom line is the genes which are associated with complex 1, particularly the complex 1, and also the proteins, they're changing as early as P6, that is postnatal day 6, much before you see any degeneration of photoreceptors. By P10, you can start seeing changes in complex 3, 4, and 5 as well, but not as much. Complex 1 proteins, each dot is a single protein in complex 1. They begin to change very dramatically. This happens much before cells have decided that they are going to die. That was very exciting, and we thought, all right, so what is changing? What we began to do was the metabolite map at P6 showed, and we began to learn our pathways. Sorry, I had forgotten until about 2 years ago. I had to go back and figure out what is pentose phosphate pathway and how the glycolysis happens. So Professor Ramesha, I remembered him, and my old Leninger textbook, I had to go back and figure out each and every. And what turns out was that many of the key metabolites in glycolysis as well as pentose phosphate pathway were altered. And we have put together, we are putting together a big hypothesis actually how this mutation from germline might be changing, and everything is altered. And this is the oxygen consumption rate that we begin to do by uncoupler and rotinone, and sort of some of the things that I learned, I relearned, and now I think I know a little bit more going back to my mitochondria days. The bottom line is the basal consumption rate in these mitochondria increases in the beginning and then goes down. Photoreceptor mitochondria have very low reserve capacity. That means if you add the uncoupler, oxygen consumption will go up only by about 20%. In most other cells, oxygen consumption goes up by 2-fold. But in photoreceptors, only about 20-25%. But before photoreceptors begin to die, they change their metabolism, and now they begin to show high MRC, very significant high MRC, which is maximum reserve capacity. So this is where we begin to think about mitochondria, and I don't think I have enough time to think about. I thought I have a few more minutes, but maybe I'll finish it quickly. This is ultrastructure analysis. I'll just very quickly mention about epigenome because it's very important in aging. A lot of people have talked about changes in epigenome and mitochondria, but nobody has really correlated that as well as we can do that in photoreceptors. And I wanted to just show you very quickly, we did a whole genome-wide bisulfite sequencing to look at DNA methylation in purified photoreceptors. And we did that with age, and we looked at differentially methylated region across the genome. And with age, guess what? The reason I'm showing you these things is we found specific changes in DNA methylation at loci that are associated with mitochondrial metabolism. So all of these data, and we did enormous amount of whole genome sequencing, RNA-seq, we have histone data, we have all sorts of other data set. Everything is leading to changes in very specific molecules associated with glycolysis. And now what is happening is with age, fatty acids that you do not utilize, photoreceptor mitochondria do not use fatty acids as substrate, but with age, they begin to use. So there's a change that happens in photoreceptors with aging that you suppress glycolysis through glucose, and you increase beta oxidation. We don't know the reason, we don't know what is happening, and this is all based on epigenetic analysis. So with age, you don't change your genes, right? Your genome is same. You're changing your epigenome, and epigenome is specifically targeting mitochondrial function, which was a complete surprise for us. And again, respiration also, same changes. This is what I was trying to tell you. There's an increased dependency on fatty acids as substrates. So I gave you 2 examples in retinal degeneration and also in aging, but I didn't talk to you about macular degeneration, complex traits and all that. But in many of those instances, pathways that are getting altered are related to mitochondria. And I had to relearn and I had to rethink about Professor Ram Sharma and Professor KURUP and go back to my days, and I said, I'm so happy that at least I know a little bit about mitochondria. I still remember. It won't take me too long, but my students have sort of now begun to look at new proteins. There are something like 100 different proteins in mitochondria that have not even been characterized. We were talking about glutaminase earlier. Mitochondria uses its own glutaminase. Mitochondria has its own sort of new type of proteins which are involved in translation. And suddenly, I find myself, almost 1/3 of my lab is slowly moving in the direction of mitochondrial function, mitochondrial DNA, epigenome, or whatever you want to call it, mitonuclear signaling. The topic, that the big thing that sort of correlates everything is that mitochondria actually seem to signal to the nucleus that it's time to change. And that happens in age, that happens before cells die in retinal degeneration, that happens in macular degeneration. And I won't be surprised if this is just limited to a photoreceptor. I think it is much more common phenomena. It's just that in mammalian neurons particularly, we are probably the first ones who have begun to explore that in such a detail. So I would like to sort of conclude with that. And so the person, people who are doing this is Coco, Anupam. Jacob Neliseri was a student in the department. He's our lab manager. He also sort of teaches biochemistry to a lot of the students, and including myself. Jimena was the one who did the epigenome study, and we collaborate with Rahul, who works with Bob Balaban, who is the scientific director and very well known in mitochondria. I find myself going to him to learn about mitochondria once again. And with that, I'm just going to, this is our group. We call it NNRL. I didn't put the name, but when I was recruited to NEI, my scientific director wanted to, because we cloned the gene NRL, which is important, he was neurobiology, neurodegeneration, and repair lab. So this is the lab. If some of you would like to join next year, I have no opening this year, but pure biochemistry. I'm now looking for folks who understand metabolism and biochemistry. We have too many people who know genetics and molecular biology and epigenome. So I don't want any of you who just know genetics or just know molecular biology. Sorry guys, I want to have people who know fatty acids, who know metabolic pathways. That's where we are going. So going back to my roots. So going back to my professors, and thank you all very much. And to all you students, just remember, life is it's a circle, sort of, you come back where you start. So I'm here in front of you guys, and congratulations to all of you, and you have a long way ahead. Keep an open mind. I think in science, a lot of us sort of get fixated into certain areas. We don't change. I think somebody said earlier this morning, you have to change with time. And if you change with time, you survive. Otherwise, you just get stuck wherever you are. So I've sort of learned one thing with time, that I need to learn that techniques should not be you should not develop techniques for the sake of techniques. You should use the techniques and tools to understand biology, and wherever they take you. With that, I'm going to stop. Thank you.
Wonderful.
In aging photoreceptor, I could see the accumulation of aged mitochondria. What about the turnover rate of mitochondria in those cells?
Very good question. We have no idea. We don't know how we will look at in mouse retina the turnover rate. We don't have any cell culture model for photoreceptors at this stage. So in vivo, mitochondrial fusion and mitochondrial, they're very hard to measure. I don't know how we will do that. If you have some good ideas, let me know. But I think we have really no idea how we will look at turnover of mitochondria in native mouse retina.
Is the threshold of mitochondria maintained?
Yes, yes. So I think that is a good part. I mean, I don't know how long they live, but each photoreceptor has about 200 mitochondria. Okay, but in rods near the synaptic area there is single one large mitochondria, but near the base of the cilia, or what we call outer segment, there are about 200 mitochondria, and in most cells there are about that much. I mean, not that many people have counted. These are very tedious studies, so some folks did once or twice and they said they don't change, so we believe them, they don't change, but there are, yeah, we don't know how to look at turnover.
Rodents are nocturnal animals, so their photoreceptors are different from diurnal animals. If so, if yes, does NRL play any role?
Very important question, which is what we are trying to. So as I was telling you, during evolution, early vertebrates had cone-rich retina. Mammals became nocturnal. When mammals evolved, all of them were nocturnal, early mammals, and diurnally evolved later on. So rather than nature, so there are two ways to think about it. Why human retina still is rich in rod photoreceptors, which are not even needed. So we don't use our rod photoreceptors most of the time. We use only cone, and cones are only 5% of retina. I think what we have in human retina or in primate retina during evolution, we decide cones utilize 5 times more energy. Each photoreceptor uses over 1 million ATPs in an hour or something like that. I mean, it just uses more ATP than anything else you can imagine. But cones use 5 times more than rods. So nature probably decided at some point that going back to a cone-rich retina will not be very good. So they created this region called fovea. So I was telling you about macula fovea. Fovea has only cones. There are no rods. So that is a very tiny area at the center of your retina, and that's where the light gets focused. So that's where the highest resolution is that allows you to see. Peripheral retina is sort of gives you peripheral vision, but primary vision comes from the fovea. And so all the cones are in the foveal region.
I have 2 short questions. One is that does it affect the mitochondria close to the cilia more intensely or early?
Yes. So very early you can start seeing by EM, you can start seeing swollen mitochondria, which suggests that there may be decrease in fusion. Okay, and the cristae within the mitochondria is sort of, you are losing that. It's no longer pretty uniform, and so we think there's a decrease in fusion of mitochondria, so the renewal is decreased very, very early stages.
The other thing is, what about the degeneration relation with the color? Does it have anything to do with the intensity of the color?
Yes and no. I mean, you know, if cones die, you lose color vision, but mice are not the good animals to look at color vision. They have only 2 kinds of cones, short and medium wavelength. For color vision, you generally need 3 for comparison of different wavelengths. So mice, we don't even know whether they really have color vision.
Okay,
hello.
One more question. I'm sure there are many more questions, but there's a cheap break after this, so we can discuss. Okay, one.
I was curious to know about the fatty acid dependence, increase in that dependency. Does it have to be, is it cell autonomous, or do serum fatty acid levels affect, like those in diabetes who have high FFA, or does it have to be something in the aqueous or vitreous humor
Okay,
levels?
there's a long answer. The short answer is we don't know. But just one thing is true, that photoreceptors are extremely susceptible to any kind of changes in their metabolism. And they do not get any substrate directly from serum. Everything has to go through what we have in between is retinal pigment epithelium, which serves as a blood retina barrier, sort of, in a way you can think of. So anything that comes to photoreceptors come to pigment epithelium, and that's very tightly controlled. So we don't know whether fatty acid transport is altered with age. We don't know whether the change is because of changes in mitochondria or availability of substrates. So this is all very, very new data, not even submitted for publication.
Further discussion
Thank you.
time. I'd like to thank